Respected chair and my dear colleagues, now I am start my topics. DNA structure and function. Myself Dr. Kartik Maiti, Assistant Professor, Department of Zoology, Raja Narendra Lal Khan Women's College, Autonomous. In 1869, Nisad discovered nuclein DNA in the cells from push, and later he separated it into a protein and an acid molecule. It came to know as nucleic acid after 1874. In 1926, Levene proposed tetranucleotide theory, which states that nucleic acid consists of only 4 nitridases. It gives 4 different nucleotides on hydrolysis. Rosalind Franklin used X-ray crystallography to help visualize the structure of DNA. This is the James D. Watson and Francis Crick, co-originators of the double helical model. Now, what is DNA? DNA or deoxyribonucleic acid is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Where is it located? Most DNA is located in the cell nucleus, where it is called nuclear DNA, but a small amount of DNA can also be found in the mitochondria, where it is called mitochondrial DNA or mtDNA. DNA structure. DNA has 3 main components. First, deoxyribose, a pentose sugar. Second, base. There are 4 different ones. 3 phosphates. The sugar, this is the sugar structure, ribose in RNA. This is the deoxyribose in DNA structure. The bases, they are divided into 2 groups, pyrimidine and purines. Pyrimidine made of 1 6-member ring, thiamine and cytosine. Purine made of a 6-member ring fused to a 5-member ring, adenine and guanine. Rings are not only made of carbon. Specific formulas and structure are not required for international. The basis, this is the pyrimidine structure, this is the purine structure, and nitrogenous bases. These are the nitrogenous bases: pyrimidine, cytosine, uracil found in RNA, thiamine found in DNA, purine, adenine and guanine. Next, DNA structure. In 1953, Watson and Crick postulated a 3-dimensional model of DNA structure that accounted for both the X-ray data and the characteristic base pairing in DNA. It consists of 2 helical polynucleotide chains. 2 polynucleotide chains coil around the same axis to form a right-handed double helix. In the helix, the 2 chains or strands are antiparallel, that is, have an opposite polarity. Backbone of each chain, which consists of alternate sugar phosphate residues, that is the hydrophilic in nature, are on the outside of the double helix facing the surroundings. The purine and pyrimidine bases of each strand face inward toward each other. The bases are stacked perpendicular to the long axis of the double helix. The base pair are 0.34 nanometers apart DNA helix. The complete turn of helix takes 3.4 nanometers. Therefore, in each helical turn, 10 bases are present. The external diameter of helix is 2 nanometers. The helix has 2 external grooves. The narrow groove is called as minor groove, while the wide groove is called the major groove. The major groove is the site for DNA binding protein. The minor group often are the site for binding small molecules. The pair of bases are always between a purine and pyrimidine, especially the pairs AT, GC, which are the base pairs found by Chargaff. There is hydrogen bonding between the bases, 2 hydrogen bonds between A and T, and 3 hydrogen bonds between G and C. 2 chains do not have the same base composition, not identical, but 2 chains are complementary, that means to each other. Such chains are called as complementary chain. This is a complementary chain. DNA is a double helix. A sugar and phosphate backbone connect nucleotide in each chain. Hydrogen bonds between paired bases hold the 2 DNA strands together. DNA strands are antiparallel. DNA strands are antiparallel strands. Double-stranded DNA hydrogen bonds. This is the antiparallel strands, purine and pyrimidine. This is the phosphodiester band. This is the 5' and this is the 3'. This is the hydrogen bonding. This is the double helical structure of DNA. And next function, nucleotides are precursors of the nucleic acids, deoxyribonucleic acid DNA and ribonucleic acid RNA. The nucleic acids are connected with the storage and transfer of genetic material, that is the concerned part of genetic information.